So last talk of the session is by Michal Sadowski from the Center of New Technologies, University of Warsaw.
Okay. Okay, hello everyone. So I'm a master's student working at the Center of New Technologies in Warsaw, in Poland. These are actually my last months as a master's student because in September I'm moving to UCLA to pursue my PhD. But this, the project I'm going to talk about will have its continuation. But before I move on to it, I would like to thank the organizers for giving me the opportunity to present my work. This is actually my first conference talk, so it's a big thing for me. Because of that, I'm also very nervous. So if you if you'll find my presentation not fully comprehensible, please find me afterwards. My poster number is there. And also this work will be published in Genome Biology within days, so you can check the paper for details. Okay, so I would like to talk about variant interpretation in the context of spatial genome organization. So there are two aspects I would like to point out at the beginning. So first of all, as all of you know, the vast majority of identified variants are located outside coding sequences, which makes their interpretation difficult. And second of all, the functional effects are usually assigned to variants through genome-wide association studies, which tell us nothing about mechanisms underpinning these associations. So advancements in the domain of spatial genome organization stands a chance to improve variant interpretation. In the reference of these two aspects, first, a part of identified variants identified in non-coding regions can actually affect genomic sequences involved in formation of chromatin interactions. And second, if this is the case, then there is a chance for a causal mechanistic explanation of variant trait association, such as, for instance, disruption of a enhancer promoter interaction. Okay, so we run our study to find out if it is frequent for genomic variants to be located in genomic structural elements, and if we can learn something about genome architecture through analyzing changes introduced by genetic variants in chromatin interaction patterns and in 3D chromatin models. So we chose to work with Chiapet data. So Chiapet may sound unfamiliar, but probably most of you know Hi-C. So the major difference between Chiapet and Hi-C is that whereas Hi-C data is represented as a heat map containing contact frequencies between elements of the artificially segmented genome, Chiapet identifies genomic segments actually forming the interactions. Okay, so yeah, but okay, sorry. the limitation of the chaplet, on the other hand, is that that one chaplet experiment captures chromatin interactions mediated only by one protein factor due to additional immun precipitation step. So you can have Ctcf chaplet, RNA pol2 chaplet, and so on. But if you look at the Ctcf chaplet, you'll see that it identifies structural features, the same structural features that high C does. So you can see chromatin loops and gatherings of those loops, which we call topological domains, or in case of chaplet, chromatin contact domains, CCDs in short. And we call interacting segments the anchors of interactions. Okay, so our approach concentrates on human normal samples. Unfortunately, there is not so much data available for different healthy individuals. So we take high quality Ctcf chaplet data for one human lymphoblastoid cell line, which is GM12878, and we map on them SVs identified in the 1000 Genome project in a population of lymphoblastoid cells. And we calculate enrichment of these SVs in Ctcf anchors and at CCD borders. Okay, so in general, we observe that Ctcf anchors are depleted of SVs found in a population, which means that they are well conserved, which is what we would expect. Okay, there are some more unexpected things happening at the CCD borders, but I don't have time to expand on that. Instead, I will concentrate on something else. So we identified Ctcf anchors occupied by enhancers and promoters. And interestingly, we observed that these anchors exhibit stronger depletion than the corresponding functional elements outside the Ctcf anchors. So, and these are anchors connected with enhancers and active promoters through RNA pol2 interactions, and the depletion is even stronger. So this suggests that that mutations in these anchors can lead to serious genome deregulations. So to test this hypothesis, we mapped on the same anchors SNPs associated previously with disease traits in GWAS studies. And having in mind the type of the cell we examine, we created two separate GWAS sets associated with hematological parameters and autoimmune diseases. And as you see, GWAS SNPs are enriched in Ctcf anchors and at CCD borders. And you can spot that the enrichment is even stronger for Ctcf anchors occupied by enhancers and active promoters compared to enhancers and active promoters themselves. So this indicates that Ctcf-mediated interactions are crucial mutational targets. The next question we asked was if chromatin spatial organization is somehow involved in variation in transcription in population. So we identified EQTLs, and we mapped them on the same genetic genome elements. So we distinguish two types of EQTLs, proximal and distal. Proximal, those located within like around 17 kb distance from correlated genes, and distal are located farther away. And you can see that proximal EQTLs are enriched in enhancers and transcription factor binding sites, which is what was shown already in the literature. But we see even higher enrichment of the proximal EQTLs and enrichment of distal EQTLs in RNA pool 2 anchors. We examined histone mark signals in anchors, around anchors occupied by EQTLs, and they show that proximal EQTLs disrupt interactions like enhancer promoter interactions at the promoter site, and distal ones at the enhancer site, which is what we would expect again. So it looks like okay. So the general picture coming from it is that Ctcf-mediated interactions create like a conserved structure, conserved genome structure, and the variation in transcription are more connected with like more dynamic RNA pool 2 interactions. Okay, and we also inspect like individual examples. So there are plenty of them, number of them in the paper. I don't have time to talk about it, but we use modeling method, restraint-based modeling method, to model topological changes after introduction of a deletion, and you can see that the arrangement between enhancers and the genes change, and these genes actually exhibit some transcription changes. Okay, and there's also we developed also web service where you can do the modeling. So coming back to these 2 points I mentioned at the beginning, genomic elements involved in shaping genome architecture are important mutational targets, and analyzing the impact of variants on chromatin spatial organization can lead to discovering causal relations between variants and disease traits. And I would like to thank all the people involved in this study, my supervisor Dariusz Prewczyński, and collaborators from the Jackson Laboratory, who do the best chaplet experiments. Actually, Ijun Ruan is one of the fathers of this method, and I'm here. I can be here thanks to the travel fellowships a fellowship awarded from RNA Society and ISCB. Yeah, thank you.
Thank you. Questions?
I think you did a really good job for your first presentation, so very nice work. So,
thank you.
I was just wondering, when you showed the last part where you were looking at the three-dimensional modeling, did you have a chance to look at high-C data and try to compare if you have contact maps which resembles any of these?
So for the modeling, we use Chiapet data because we need because this is restraint-based modeling, and as a restraint, we use chromatin interactions. Okay. So if you call loops from high-C data, you can use them for the modeling. Okay, but we need the actual interactions. Actually, like low resolution models can be built with our modeling method based on high-C maps, but you won't see there like individual loops. Okay, you will see like a general chromosome structure, let's say.
So, are you taking nearest neighbor approximations on this? Is the underlying model based on nearest neighbor approximations?
No. This is based on Monte Carlo simulations.
Okay.
Yeah. Right. Yeah, but we also explored methods using this and using MDS for optimization. But this is I think this works like using these models, you can actually see the individual loops. So this is
I understand.
This kind of maybe better.
Thank you.
Okay. One last question here.
Yeah, I had a question. So, how many of your GWAS sites are in enhancers as opposed to just in Ctcf parts of loops?
Sorry, can you?
So you have it's well understood that a lot of GWAS sites are in enhancers, yeah. But you're also describing that some of them are in Ctcf binding sites, which may form loops independently of enhancer status.
Yeah. Okay. So we didn't check what's the proportion. I mean, because okay. You mean that a part of SNPs that target let's say that intersect Ctcf anchors will also intersect enhancers, right?
Right, but it would be interesting. I mean, it's pretty well studied. A lot of GWAS sites and enhancers. It would be very interesting to study if there's just a three-dimensional conformation effect just by Ctcf sites, which isn't just an enhancer-mediated or enhancer-located effect. Mm-hmm. It would be an interesting thing to look at.
Mm-hmm. Yes, thank you for this comment.
Okay, let's thank again Michal for this kind of presentation.
Thank you.
So yes, as closing remarks, I would like to make some announcement on behalf of the organizing committee. So first, just to remind all the presenters, also the poster authors, but in general, everyone interested in publishing original research in a special issue that the VarI has been organizing in the last years. Last year was in BMC. Probably this year we will change of journal. This is under discussion, so you feel free to send suggestions. One possibility would be NAR, genomics, and informatics. Also, you are all invited to actually get engaged into the organization of the VarI. So this is an open group, so anyone interested, please contact us. We will be happy to have you on board. Also, if you have any suggestion about the general organization of the course, or you have suggestions about keynote speakers for next year, please, again, drop us an email. Also, we would like to invite you, of course, for next ISMB at Montreal in 2020. That will be the 10th VarI session, so it will be a special year. and also to thank you for your participation this year. The COSI will continue now at the poster session. If any one of you is interested in going for dinner, also let us know. You can send us an email or just come after the session. And let's thank again all the speakers and presenters today.